So welcome to everybody. First of all, I am Youri Uneau, and she is Maéva Marcin. So we are from Guadalupe, Assofri. So first of all, I will present you Assofri because I think that nobody knows us. So Assofri is a small farmer association. So we have 60 farmers, 60 members for almost 300 hectares cultivated of vegetable and fruits. And what we do in Assofri is provide agricultural advice to our members. We also have an experimental station where we did the experiment in organic agriculture, and on our plots we do training, experimental research, and we do sensibilization of the general public on sustainable agriculture, and we also do some direct commercialization of our products. So next. So our local partner are all the organization in agriculture and project about sustainable agriculture. So I will talk about citrus culture in Guadeloupe. So citrus culture is a small crop, but important crop because it's economically important and culturally important for Guadeloupean people. It's the third largest fruit production after melon and pineapple. And after the discovery of HLB, as you can see on the graph on your right, there were a big decrease of production depending on the different varieties. And as you can see, oranges, mandarin are very sensitive, and there is a very big decrease of production. And 2 years after the discovery of HLB, we saw a loss of production between 50 and 90%. So it was a big impact on our growers. And in 2020, there is a decrease of half of the production area in Guadeloupe. And now growers are waiting for more tolerant and performing varieties to restart production. So after the discovery of HLB, we did some analysis of infection of all the plots, of the citrus plots in Guadeloupe. And what we saw is that the entire territory was infested by Candidatus leberibacter and by the Asian psyllid. So we tried to do some sanitation with the politics, but it wasn't so effective. So since 2016, there is no sanitation, and now we have to learn how to live with the disease and find new solutions.
Okay, so our main experiment on the Tropicsafe project is a field evaluation of varieties of citrus and practices. So we compared and evaluate 11 citrus varieties and 4 rootstocks. We used diploids and tetraploid rootstocks because the tetraploid are expected to be more tolerant for HLB. And for that, we have 4 plots located on different sites that we monitored during 5 years between 2017 and 2021. And we used 2 different managing strategies for the plots, the IPM, integrated pest management, and the organic management certified by EcoCert, the second one in green. The only difference that there is after 2019, the only difference that remains on both strategies is that for IPM, the fertilization is mineral and is more often. And there is foliar and soil fertilization. And in organic, it's only organic fertilization. So let's introduce the experimental system. So this is the Guadalupe, and we have the 4 plots that I said. The 4 plots are 2 trials replicated. So the first one is an orange trial where we planted on one plot is 162 trees planted on 2015, where we tried 6 different varieties of oranges grafted on 3 different rootstocks. One is in Asofui, this is the green row, managed in organic management, and the other is at a producer member of Asofui in Capester. No, c'était bon. And the other trial, replicated twice also, is small citrus. We say small citrus essay, but small citrus is for mandarin and tangelo varieties. One in Asofui again, managed with organic management, and the other one is in a Sirad station in Capester. This one, 5 different species of tangelo and mandarin was tested, and there is 90 trees on one plot. And here are the pictures of the here in Asofui and there in Sirad. So during those 5 years, we monitor the plots, and we collected the data about psyllid, the number of psyllid that we observed on the trees, the general aspect of the trees, mortality, the growth of the trunks, the presence of Candidatus Liberibacter asiaticus with the PCR length method. That means that we tested all the trees to know if they were positive or not to HAB, and the chlorophyll index, and just for one experimentation, the bacterial load that I present then. So the first results we had about the varieties and rootstocks is this result. You can say for the small citrus trial, the first table, we have the varieties first and then the rootstocks. So we selected some criteria to evaluate the varieties, and we could say that for the Tangelo, it's the Tangelo Jackson that was the more interesting for us, and in the Mandarin is the Mandarin Temple for his productivity. About the oranges, we found that the Valencia Road Red variety stands out. But for the rootstocks, we didn't find a significant better performance for tetraploid rootstocks. But in Sirad, they had better, more interesting results. So it could be interesting to go further in that experiment of rootstocks. And we need to continue to analyze our trees because they continue to grow. And so now comparing the plots, so the practices, we have some interesting results. Like the first one is about pressure, psyllid pressure, and HLB pressure infection. At the end of the experiment, you can see that almost all the plots were 100% infected by HLB. And we also observed on the three sticks represents the percent of the infection on each plot. And the 3 last, we observed a lot of psyllid there. And in 2 or 3 years, they were already 100% infected by HLB. But the first one, which is the plot in our farmer member of Asofui, it's a plot in altitude. It's 400 meters, and we didn't see psyllid there. We only observed 2 psyllid in 5 years, but yet it reached more than 64% of infection by HLB. So it brings the question of a potential alternative host vector. I mean. But we tried to find it, but we didn't find nothing. And another result is about the rate, the mortality rate at the end of the experiment, means the number of dead trees at the end of the experiment. You can observe the sticks are the different plots, but we have a high number of mortality on almost all our plots, unless this one, the little stick, which is the CIRAD station plot, the plot with a plot that is in CIRAD station, and this one is the only one where irrigation and fertilization is very well controlled, unless the others. So we can conclude that when the trees are infected with HLB, it's very, very important that there is no stress factor. So no water stress and no fertilization stress. So in our experiment, the IPM management appears to be the most favorable. Another practice experiment that we have done is a test of biostimulants, and we had a very interesting results in that trial. So we did it on one plot that we separate in 3 modalities. In one block, we sprayed a commercial biostimulant named Green Flash. In another, we sprayed natural manure that we've done with this plant named Patagon, which is a medicinal plant. And the other block is the control block where we spread no biostimulants. We did that for 2 months, and we spread the biostimulants each 2 weeks. And during that experiment, we measured the chlorophyll index and the bacterial load. And what we found was very interesting. About the bacterial load, we measured it only on one variety, it's the Tangelo Nova, and we found it that the one of the middle is the control one. Each stick represents time. So the first is the bacterial load at T0, the other at the middle of the experiment, and the third at the end of the experiment. And we can see that the bacterial load increase and decrease for all the modalities, but it stayed very lower at on the modalities where we put when we spread biostimulants. So there is a possible biostimulant effect. So it would be very interesting to go further in that research and to experiment, to expand the experiment. So all that knowledge we are transmit by a lot of communication events, technical days, trainees. We also edit documentation videos that you can find on our sites. That's it.
And, to conclude about this project, our perspective after all this work is to go further in the varietal and varietal evaluation, and to be more performant in the practices, linking with some type of fertilization like fertirrigation some type of pruning to restart growth, biostimulation. As we shown, hydric stress is very, very bad for trees under HLB. So we have also to find more tolerant species to hydric stresses, and maybe more innovative water efficient systems to reduce all these stresses. So we want to thank all the teams, especially Assunta Bertaccini and our partner from CIRAD, and all the participants, and thanks for your attention.